In this lecture, we're going to talk about mutations. Now, typically, when you have meiosis, everything goes as planned. Typically, nothing bad happens. However, when meiosis goes wrong, mutations are created. So the majority of the time, meiosis occurs just as it's supposed to. However, boo-boos happen. And when I have a boo-boo in meiosis, I now call them a mutation. Mutations are simply changes in the genetic code. I've messed something up. I've swapped something. I've put something where it doesn't belong. I have had a crossing over that just didn't work right. Something has occurred that has changed my genetic code. Now, this can have several different effects. Sometimes mutations can be neutral. If this is the case, then the mutation has little to no effect on the expression of a gene. When we get into DNA and protein synthesis, when we take our genetic code and we figure out what part of it gets used, and then we translate that into an amino acid sequence, you'll find out that we have 20 different amino acids. Different genetic codes code for the same amino acid. So sometimes a mutation can occur that will change just one letter. However, it changes it to a letter that still works for the amino acid that it needs to have in that code. So sometimes mutations can have little to no effect on the expression of the gene in question. You won't even know that it has happened. Sometimes mutations can be beneficial. When you have this, if a mutation is going to be beneficial, then it's got to increase something about that gene. So in these, it increases the genetic variability in a species, which allows for increased survival in a changing environment. So it increases my ability to survive. It increases my chances of survival. So it is a beneficial mutation. Now, on the flip side, if it can be neutral and it can be beneficial, mutations can also be harmful. So you only have three options. It's either neutral, where you don't see any effect from it. It is beneficial, so it helps you survive, or it is going to be harmful. These are most often lethal to a zygote, which means that the zygote will not come to fruition. The body would spontaneously abort it. If the zygote is allowed to mature, the other side of the coin is that it is the typical cause of many genetic disorders. And we will talk about a lot of these in class when I see you. So you have three options: neutral, beneficial, or harmful. Now, mutations are typically caused by one major thing. The most common error that causes a mutation is non-disjunction, which just by the name you know something doesn't get separated. Something fails to separate the way it is supposed to. They stay joined rather than getting torn apart. What non-disjunction is when homologous chromosomes fail to separate during meiosis 1. So anaphase 1 doesn't go as it is supposed to. So I'll put this down in parentheses. When I go to separate my tetrad, when I break up my homologous pairs, something doesn't happen and it does not split the way it's supposed to. And that would occur in meiosis 1, specifically anaphase 1. What this does is it creates an abnormal number of chromosomes in the gametes at the end of meiosis. So instead of ending up with just one copy, one set of my chromosomes in my gametes, I'm going to end up with something different, something that is not the way it's supposed to work. Remember, I always get 4 gametes out of it. My total is always 4 haploid cells. So an example of this, 2 gametes get extra copies of the chromosomes, while the other 2 get none. In the picture that I am now showing you, that shows non-disjunction. So in that middle picture where I have anaphase 1, if anaphase worked out the way it is supposed to, I should have 2 sets of my sister chromatids on, say, the south pole, where instead in the picture I only have 1 of them. So I had non-disjunction. My tetrads did not split apart the way they are supposed to. My homologous pairs didn't get split up. 1 set of my homologous pairs stayed together, so the whole tetrad went to the north pole of my cell. In the end, when I get through meiosis 2 and I'm through to my 4 haploid cells that I'm going to create, I end up with an extra copy of a chromosome that I'm not supposed to have, instead of having it equally split. So 2 of the top 2 of my haploid cells in this picture have that extra copy, whereas the bottom 2 cells did not get it. So they are 1 chromosome short. If a zygote is allowed to mature, the body doesn't spontaneously abort it, a genetic disorder usually will present itself in the organism. In the next class, I will talk about several different genetic disorders that happen when non-disjunction, or when something, one of the other mutations that I'm going to talk about, when one of those happens. But the most common error that you come across is going to be non-disjunction. The other ones, there's other kinds, and I'm going to tell you what they are, and I want you to define them. I want you to tell me what their definitions are, and then we will talk about them in class. One of the mutations you can have is called an addition. If I have an addition, I have a deletion as an option. I can have a duplication mutation. I can have an inversion mutation. And the last one that we will talk about is called a translocation. All of these are options. So along with non-disjunction, which is the most common, I can also have addition mutation, deletion mutations, duplication mutations, inversion mutations, and translocation mutations. Try to come up with your own definitions, and we'll see how good you are when you get to class. And then that is where I will start when I see you.